Synthetic Biology by Michelle Behringer and Alexis Gomez. What comes to mind when you think of synthetic biology? The term synthetic biology was coined by Barbara Hoban in 1980. It was used to describe scientists' manipulation of living systems through the use of recombinant DNA to genetically engineer bacteria. In other words, synthetic biology was taking a gene from one cell to another using a vector. But in 2000, at the American Chemical Society meeting in San Francisco, synthetic biology was described as constructing completely new systems or organisms that are not found in nature. Cells are no longer living things to be manipulated, but little machines to be created. The biological parts necessary to create these new species involve DNA sequencing, parts that are combined into devices. These species are then able to perform more complex functions. A recent example of an engineered complex function is the transformation of a sugary seaweed into ethanol, a source of biofuel. The problem with this seaweed is that it contains sugar molecules such as mannitol and alginate, which are difficult for microbes to ferment into ethanol. Scientists of the U.S. Advanced Biofuel Company were fortunate to find the bacteria Vibrio splendidus. Vibrio splendidus is a marine bacterium which obtains these genes necessary for alginate lysis and the fermentation of mannitol sugar molecules. Over 20 genes were used from the Vibrio splendidus bacteria in sequence with the genes of E. coli, with the addition of a fermentation pathway and the deletion of some E. coli genes. This new bioengineered E. coli is capable of fermenting mannitol better than the conventional yeast, as well as fermenting alginate into ethanol. Seaweed has become increasingly popular as a new source of biofuel due to the reconstruction of E. coli.